So this part of the review session is just going to go over ionotropic receptor function. There's three major ionotropic receptors that we want to compare, just because the comparison between the three kind of tells us a lot about what they do. So real quick, I'm just going to say we've got AMPA receptors, we've got NMDA receptors, and we've got GABA A receptors. And we're going to compare AMPA and NMDA a lot more later on, but just overall, one of the similarities between these is that they're both heterotetramers. What this means is that they're both composed of 4 different subunits. So the subunits within an AMPA receptor are often called like glue A whatever number, right? And then the subunits for an NMDA receptor are glue N whatever that is. So by being a heterotetramer, an NMDA receptor, for example, is made of 4 different types of glue N subunits, whereas an AMPA receptor is made of 4 different glue A subunits. GABA, the GABA A channel, is actually going to be heteropentameric. So whereas tetra means 4, penta means 5. So this is going to be a channel composed of 5 different GABA subunits, or GABA receptor subunits. The specific names of those, definitely look at them. We're not going to ask you those without talking in some way about function, right? But you should know that for example, an AMPA receptor is going to be made up of glue A subunits, whereas a GABA or an NMDA receptor is going to be made up of glue N subunits. Just things like that. Very broad strokes kind of definitions of what the subunits are. Please don't memorize the specific number of the subunits. There's only one exception to that, which we'll talk about. But again, these AMPA and NMDA receptors, they're both cationic. And whereas AMPA receptors are passing only monovalent cations, meaning one charge, monovalent cations, we can also say that these are selectively cationic. NMDA receptors are essentially non-selectively cationic. Where this becomes important is the fact that NMDA receptors can also pass calcium, and that's going to be really important for our discussion of synaptic plasticity later on. GABA A receptors, well, obviously, they're going to be anionic, so those are going to pass chloride. Structurally, there's definitely things we want you to think about. Unfortunately, there's not much to it besides just kind of rote memorization. I can kind of see if I can try to simplify things a bit. So your AMPA receptor is going to contain these 4 domains. You're going to have, so number 1 is going to be your amino terminal domain. And that's just going to be referencing the fact that any receptor or any protein really is going to have an amino terminal domain and a carboxy terminal domain, just based on how you're putting those peptide monomers together. So you're going to have an N amino terminal domain. You're going to have a ligand binding domain, which they usually draw kind of looking like Pac-Man for some reason. So it's going to be your ligand binding domain. You got this transmembrane domain. So number 3, transmembrane domain. And then you have a carboxy terminal domain. So this is number 4, carboxy terminal domain. And again, this is just one of those subunits. So an AMPA receptor is going to be built of 4 of these. So right, each of the 4 subunits of an AMPA receptor has an N-terminal domain, it has a ligand-binding domain, transmembrane domain, and a carboxy-terminal domain. So when we talk about how many glutamate molecules can bind to an AMPA receptor, the answer is technically 4. The reason we often say 2 is that it requires 2 to bind in order for it to do anything, but it can bind up to 4, and by binding more neurotransmitters, it actually increases its conductance to, again, sodium and potassium, those monovalent cations. This is most similar to our kainate receptor, to the point where people will often just say AMPA/kainate. The names of these, so AMPA receptors, kainate receptors, and NMDA receptors, those are actually based on what are called agonists. An agonist is something that opens the receptor. So before they ever understood why, or like what AMPA receptor opened in response to in the nervous system, they knew that it opened in response to AMPA. So that allowed them to say, okay, well, we know that AMPA opens this channel. We know that this molecule called AMPA opens this channel. What is the endogenous neurotransmitter in the brain that most closely resembles AMPA? And the same was true with kainate, and the same was true with NMDA. They had these 3 different agonists for what they thought were 3 different types of channel. Well, they are 3 different types of channel, but what they found out later on is that the endogenous neurotransmitter that opens all of them is actually glutamate, and their differences in responses are actually based on the structure of those channels, and things like the magnesium block, which we'll talk about. So that's why they're called AMPA receptors. That's why they're called NMDA receptors. Same is true with acetylcholine. If you ever see anyone talk about nicotinic acetylcholine receptors, those are named in that way because the original experiments to learn like what are they opened by found out that nicotine was actually an agonist for those receptors. It wasn't until later on that they figured out, okay, these are actually endogenously opened by acetylcholine. So we talked about AMPA. Let's kind of go over NMDA real quick. In terms of the really nitty-gritty structural details, we're not expecting you to really get into the woodwork with this. We know that NMDA receptors require binding of glycine to function. This sounds really important, and it is. The thing is, though, glycine is pretty apparent in most cells. So it's rare to see a condition in where an NMDA receptor isn't bound to glycine. It's kind of a convoluted thought process I just went down, but what essentially I'm trying to say is that a lot of times people think that because glycine binds to NMDA receptors and because it's required, it's essentially guaranteed that it's going to be there. And of course, that's an oversimplification. I'm sure there's conditions in which glycine isn't there, but the fact that glycine is necessary isn't as important as it sounds. Glycine is just kind of always around, so it happens to always kind of be bound to these NMDA receptors. That's actually why we say that these are gated by glutamate, is that even though glycine is necessary, the availability of glutamate is more on an activity-dependent basis, meaning glutamate release is what usually gates these open or closed. In fact, they're usually bound to glycine, so you wouldn't consider glycine the transmitter, because if it was, then they would always be open, essentially. I hope that makes sense. The major structural thing we want you to care about for this, again, they're going to have an amino-terminal domain, they're going to have a ligand-binding domain, they're going to have a transmembrane domain. The huge, really important piece to this, though, is that these NMDA receptors have a magnesium blockade. And this leads to a lot of confusion because the channel opening is ligand dependent, right? You need glutamate to open these channels, but current flow is actually dependent on voltage. So just to say that the channel opens, and this is kind of a counterintuitive idea, but just to say that the channel opens isn't enough to say that current is flowing through, because the channel could be open in a sense. Structurally, it appears open, but there's something still in that channel pore that's blocking the flow of current. And what that's going to be in this case is the magnesium ion. So this magnesium blockade establishes a voltage dependence on flow. Whoops. And what does this mean? Like, why does this matter? Well, as it turns out, most glutamatergic postsynaptic neurons have both AMPA receptors, call this AMPA, and I'll draw this in a different color. Let's call this red, NMDA receptors. So you've got both AMPA and NMDA receptors. And these receptors are both gated by glutamate, right? So again, glutamate is blue. So here comes our glutamate binding to our AMPA receptors, needs two of them to bind in order to cause influx. And the ions that AMPA receptors influx, I don't know why I changed from blue, are going to be sodium and then a little bit of potassium efflux, but we know that because these are sitting at the equilibrium potential for potassium, that's not going to be as relevant. Your NMDA receptors are also going to bind 2 molecules of glutamate or even more. I think because it's also a heterotetramer, right, there's going to be 4 ligand binding domains, yada, yada, yada. So we've got all our glutamate out here. But then that magnesium blockade is going to prevent the flow of sodium and calcium. Remember, these NMDA receptors can pass calcium, and that's what's going to make them so crucial for synaptic plasticity. So these are non-specifically cationic. So what does this mean? Well, once enough sodium influxes to sufficiently depolarize this cell, that magnesium blockade is actually going to pop out. And that's what we mean when we say that the magnesium blockade establishes this voltage dependence on current flow, is that if the inside of the cell isn't relatively depolarized to a certain threshold, that magnesium is perfectly happy sitting in that pore. But we know that positive charges repel each other. So eventually, once you build up enough positive within the neuron, all this positive within here is going to repel this positive magnesium. So what that's going to lead to is this magnesium goes away. Oops. This magnesium blockade goes away. And all of a sudden, you have a conductance to calcium. And we say that this calcium conductance is voltage dependent. Another way to say that is you need a pretty strong depolarization of this postsynaptic neuron in order for calcium to come in at all. And this will become apparent when we talk more about synaptic plasticity, but essentially what I mean to say is that calcium conductance, because it's so crucial for synaptic plasticity and because it's so crucial for strengthening the synapse, we don't want it to strengthen any old synapse that has a baseline amount of activity. We want to only strengthen the synapses that are receiving a ton of activity. So this is a way to mediate that activity-dependent strengthening of synapses that we see in synaptic plasticity. And that's going to be pretty core to this idea of what synaptic plasticity is, right? It's a response of the postsynaptic cell, or a response of the presynaptic cell even, but in this case it's postsynaptic. A response to an increase in activity that's going to lead to an increased sensitivity of the postsynaptic neuron to glutamate. And we'll talk about that a little bit more as we talk about synaptic plasticity. But essentially what's going to happen, give you a little preview real quick, is that calcium influx is going to lead to some kind of intracellular cascade that eventually leads to the exocytosis in the postsynaptic neuron. Oops. I'll just write it out. Leads to exocytosis of AMPA containing vesicles. So I want you to think about what this might do to the sensitivity of the postsynaptic neuron to glutamate. And we'll talk about that in the next video too.